Thanks for the introductions and thanks for inviting me to share some of our work here today. So before I start, I want to introduce you some of our co-authors. So Stacy and then Harry and then Ali. And Stacy and Ali are more genomic people. They actually provided more gene editing of this one. So I prepared a number of different topics. My topic today is really a summary of Canadian forage breeding. It's really 100 years of history. So it's going to be a perennial breeding in some of achievements and some of future perspective I'm going to talk. So I really outlined my presentation in 4 sessions. One is kind of summarize all the hard work, great achievements over 100 years. And then I would talk about recent varieties, maybe recent 5-10 years varieties, which one have been released. Then I will give you some idea of what you're going to expect next 5 years from Canadian breeding programs. And then some of the new technology, people are using it, and then including gene editing, which is very popular now, and then maybe controversy in certain aspects. So before I start, many of you know the forage seed itself. It's really important for forage varieties. Forage variety basically support forage seed industries. And you can see that this is kind of some of statistics, around $130 to $180 million seed export every year from Canada. It's from Peace region, Saskatchewan, and also Manitoba and some other smaller areas. We also have domestic large consumptions. We have 11 million heads of beef cattle rely on forages, 1 million dairy, and then some domestic hay market, and probably also emerging hay market to Saudi Arabia or Taiwan or a number of other countries. But also wants to bring you up to this lower corner, the bison industry. And it seems very interesting industry. And a couple years back, bison seems smaller industry, but it's one of the fastest growing industry. And I can see they are really demanding forage informations, forage seed informations, and then different forage species. But all of you probably know forage by far the most important crop for land management. And I know when we're talking about perennial forages, people try to say this is not really cash crop unless you're talking about forage seed. But forage itself is really important for all this cash crop, right? We crop rotations, building the soils, and you can see soil organic matter and nitrogen fixations. And then especially when we have a dry and wet cycles every year, you probably see more salinizations and more marginal land, and then forage really is important for this kind of environmental good and service. So probably all of you know all these things. So I want to give you some idea of 100 years of forage breeding. So I looked for, when I'm preparing for these presentations, I went through a number of different documents. A majority of them are Agriculture and Agri-Food Canada publications. So majority of forage breeding programs, they were established around 1920s. One of the program at the university is University of Saskatchewan, 1920-22. Then they did probably 20 years of breeding and moved to Agriculture and Agri-Food Canada, Saskatchewan. So I looked at these 11 research centers in Agriculture and Agri-Food Canada. And then the reason I brought it up here is all of them at least released 1 or 2 varieties. Some of them are many varieties. So it's just not the name, it's really developed varieties. There's 4 universities, University of Saskatchewan, University of Manitoba, Dr. Takatani is here today, and University of Guelph and University of McGill. But I also want to highlight here is up into Beaver Lodge, AFC only have 4 breeding institute, the breeding programs actively today. So the rest of them are really more like a history now. And then university is only 2, University of Saskatchewan and University of Manitoba. So it's really shrinking or downsizing if you talk in a precise way. So there are many people. This pictures doesn't really represent everybody, but I give you, this is kind of idea who they are. Over 100 years, many forage breeders worked in Canada. In Eastern Canada, I wasn't able to bring former breeders. First one is Dr. Robert Knowles. Some of you probably know he's one of the best grass breeder in Canada. Dr. Surya Churiya from Lethbridge, who has retired. Dr. Bruce Goldman, who has also retired. And then this five of them are forage breeders, and Dr. Nitya Kanal also do some germplasm improvement. So Annie Klassen is a Quebec City, and Dr. Yusuf Papadopoulos and Dr. Doug Katani from University of Manitoba, and Dr. Harry Powdell from Lethbridge AFC, and Sean Aslin from Swift Current. So Sean is slightly different from other groups because his focus is more on native plant breeding. So there are many people involved in this over 100 years, technical staff, entomologists, and also pathologists, and importantly, lots of important technical staff. So I counted 254 varieties. This one doesn't include ecological variety developed by last maybe around 1990 to 2005, or around that time, because it's a very different system. This doesn't include turfgrass varieties. It's all forages, and we have 154 varieties and then 25 different species. So this is kind of current locations: Lethbridge, Saskatchewan, Swift Current, University of Manitoba, Quebec City, and Kentville, Nova Scotia. This is now more active breeding programs. So I give you some summary of which species, and then whether we have active breeding programs or what's going to happen for some of species. So Alfalfa by far is one of the top species developed for varieties. You see 31 varieties since 1955 to 2019, and I'm really not counting everything. It doesn't include any introduction from other country. It's all Canadian bred varieties. And you know probably beaver alfalfa, Anik, it's a yellow flowering alfalfa. Sandvine, there's 4 of them, Novan, Mountain View, Sisomioquest, there's 3. Red clover, there's 13. And then number of institute worked on red clover, Eastern Canada, majority of the Eastern Canada, Beaver Lodge, Birdfoot trefoil, and also Alsac clover. So based on the record, Alsac clover was only developed in Beaver Lodge research stations. But I know right now, Alsac clover is some degree in Eastern Canada by Kentville research stations. And alfalfa, if you look at this active program in Canada, 1+ sign really means 1 breeding program. But the sign of working is maybe different, some really smaller scale, but doing something. But alfalfa, there's 5 active programs. Birdfoot trefoil, probably 1. Alsac, it's maybe 2, maybe 1. But sweet clover, there's no breeding programs. So now comes to grasses. We have 90 different varieties over 100 years. And then smooth brome grasses, they're mainly in AFC, Saskatchewan research centers. And somehow, since 1989, there's no new variety released for Western Canada for smooth brome, and then no active breeding program. So you potentially expect not much variety coming up for smooth brome. Meadow brome, there's 1 hybrid brome. It's basically my program working on this. Timothy grass, there's 19 varieties, and you know Climax and also Alliance, 3 breeding programs working on it. Orchard grass, 15 varieties, and some of the varieties you probably know, and then there's still 3 breeding programs. Crested wheatgrass is really downsizing compared to its historic release. Not many varieties, maybe basically my program work on crested wheatgrass, but it's very hard to get funding to crested wheatgrass and keep going. So we really hope still keep smaller scale breeding. Intermediate wheatgrass, there's 2 varieties, Chief and Clark, but you probably hear from Dr. Doug Katani today, it's also more shift their direction to perennial green, and that's probably a good thing. Hybrid wheatgrass and tall fescue and creeping red fescue. So creeping red fescue is interesting. We were talking about, is it forage variety or is it a seed variety or for turf? So I included here as a forage because it seems like it released as a forage, but boreal from Beaver Lodge. I couldn't see any active breeding in Canada for creeping red fescue. So there are also a number of minor forage species. If you, probably some of you really know all this wild rice, right? Russian wild rye, Alta wild rye, and Buharan and tall wheatgrass. So all these 4 species are only developed in one research station, that's Agriculture and Agri-Food Canada, Swift Current Research Centers, and then there's no active program. So you probably wouldn't see or expect any new varieties for this species unless there's a new funding and people wants to revisit the germplasm. Pubescent wheatgrass was 1966, green leaf from AFC Lethbridge, and the reed canary grass, there's still 2 active program, University of Manitoba and AFC Quebec Center, they are doing some reed canary grass. Ryegrass, there's one, Norlia, it's from Ottawa. Native wheatgrass, there's not recent release, but they have been working on this species a few times. Slender wheatgrass, I know it's quite popular seed varieties, but there's no active breeding for this species. So all these red ones, there's no more breeding, and so that's a thing. So many of you probably know how forage varieties have been developed, but I give you some very general saying. So first, really, forage varieties, you have to bring the germplasm from Canada or different world, different collaborations or somewhere in the gene banks. And then from there, you potentially grow a seedlings, a couple weeks in a greenhouse, then take it to the field. And then in the field, all individual plants, they have an ID. So you keep this ID for a couple years and then keep evaluating and eventually select plants. And then legume crops, you can use cages or tunnels to using a bee to pollinate them. And grasses, you can use a greenhouse or isolated area. And in recent years, this grass pollination tent you can see from the bottom, this corner it's released, developed by a company in the UK, and it's really pollen-proof, and it really helps lots of things. It's cheaper once you have it, and produce way more seed than a greenhouse. So that's kind of general how we breed, initiate grass breeding, but now probably this is more important to how many years we really need to consider before the variety comes to the actual grower's hand. So as we mentioned, the first one, line development or initial germplasm development takes at least 6 years, maybe even more if you do a couple cycles. Then you do the field testings and initiate in one locations, and whichever is really looking promising for certain trait, you can bring it to other different locations for testings. And then once you have all this data, you can release a variety. After 2014, you don't require merit testing, so that really means you can provide a really good description of variety and you can register, which is not a great thing, but that's how it works in the regulation world. So after this variety is actually released, you have 2 years to develop breeder seed, another 2 years to foundation seed, another 2 years to certified seed. So it almost takes 20 years. One variety actually comes to a grower's field. So that's how it works. So the one thing is we really have to more strategic or visionary, and an industry involvement in this is really critical, because that's how you're going to decide 10, 20 years thing. So that's kind of overview of the history and then breeding overall. Now I give you some of the varieties released in last couple years. And the first one is AEC Truman alfalfa. This was released by AFC Kentville in 2018 by Dr. Yusuf Papadopoulos. This one is really released for rhizomatous. So alfalfa has a rhizome or taproots, and winter hardy. Its interesting characters is it's also drought tolerant, also waterlogged intolerant. So that's kind of two very different trait, and so this seems a very interesting, good variety to the industry. So we also released in University of Saskatchewan a number of varieties. A hybrid brome, it's a torque. It's really released by Dr. Bruce Coleman. It's a high forage seed, high forage yield. And also another meadow brome in 2020. It's also towards more grazing or beef industry. So this torsion is more grows in the fall, so people can use for grazing or stockpile grazing. So one of the first variety, Timothy grass, there are a number of varieties released in Eastern Canada, but till now it's the first one released in Western Canada. It's a quite tall varieties, very high seed yield, and also high biomass yield. So in 2020, also released the hybrid wheatgrass. It's a hybrid wheatgrass. It's really hybrid between two different species. Because of that, the seed yield tend to lower. So we targeted high seed yield. And this variety, it's really for sealand reclamation. So we also worked on salt tolerance and reducing a rhizome. Because hybrid wheatgrass has a really strong rhizome because of the parental species, quackgrass. There are other varieties, Nikong alfalfa from Quebec, Prestige Timothy from AFC Quebec, Mountain View Sandfoin and as Glenview Sandfoin from Lethbridge, so they are more towards growing sandfoin with alfalfa field. So what's new in 2025 this year? This is probably more interesting to some of you. So we're going to release one turfgrass. It's a forage type turfgrass. Turfgrass and overall in Canada, it's a winter hardiness. We call it marginal in most cases. So this is really working to develop high winter hardiness and also good forage characters. So Dr. Yusuf Papadopoulos mentioned EFC Kentville going to release acid-tolerant alfalfa. We all know alfalfa is very sensitive to acid soil, right? So it's acid-tolerant alfalfa. And Harry Powell mentioned about going to release the grazing-tolerant alfalfa. But maybe he will talk about this grazing-tolerant alfalfa in his presentations. There's one orchard grass. It's also from AFC Kentville. Maybe it's another interesting thing is Dr. Doug Dunny from Manitoba is going to release a perennial green intermediate wheatgrass, which is also another interesting one. So what to expect in the next 5 years? So we've been working on a number of things. It's on the horizons. In alfalfa, you probably see a high sugar content alfalfa. It's more soluble sugar content in the stems, and then the AFC Quebec groups have been working on this for quite some years now, and then they were in a testing stage for varieties. Reed canary grass, maybe next couple years, you probably see from University of Manitoba releasing a reed canary grass varieties, sandfoin, more persistent, hardier type of sandfoin, and alfalfa for yellow flowering. So it's falcata type or yellow flowering wasn't really the mainstream of alfalfa. Maybe some companies probably doesn't like it because it persists too long or forever. But there's some niche market to this, and then we've been working on this. So hybrid brome, potential one, and then another salt-tolerant alfalfa coming up soon. So my final portion of my topic is new technologies. And then we've been doing this conventional for 100 years. Maybe some changes are needed, or maybe some different aspect of thinking of this breeding should be applied in these breeding programs. So these are people still working on drought. Drought has been a big topic in the last 5 years. People are working on red clover seed yield, which is probably related to seed industries and acid tolerance, high sugar content, lipid content. When the greenhouse gas emission is important thing, people start to think about lipid content. So it's a lipid content, high seed germination of Sicer milk veg, salt tolerance, and then switchgrass, which is one of the bioethanol native grass. Quebec group has been working on it, and then Timothy and various maturity. So one of the technique, I really think it's very applicable to agriculture overall, especially in the breeding, it's a drone. A couple years back, we had a summer student and who turned to a graduate student, and we were talking about drone, and that's about 7 years ago. And then even myself, I really don't understand enough. I was thinking drone is probably taking a couple pictures and maybe developing good videos. So drone can do tons of different things in the research. We know other applications, but in these examples, this is part of my breeding programs. So the drone can have different cameras. In this case, it's a multi-spectrum camera. You can use hyperspectrum too. So it can only takes, so one example here is in the center are some mosaic map. So this is one breeding nursery I have. If I do myself, it really takes maybe 1 day or maybe slightly over 1 day to complete all the individual plant ratings. This drone only takes 4 minutes. And then that really means we can do number of times, again and again. And then it really can develop into actual plant. So you can see from mosaic map to different software, it comes to actual plant. It can give you green area. It can predict your forage biomass. It can give you a plant height. So it's a very useful tool. So here's one example. So this is a prediction of forage yield of Sicer milk wedge. First one is one of our graduate students worked on. Just the most important trait was a stem length. So it's a taller stem, predicting a higher biomass. So you can see in about 69% accuracy for that if you directly mirror it by stem. But in a drone can predict 92% accuracy and based on a number of readings for forage yield. So some other later stages are not very accurate because stockpiling, it's October, it's already yellow, and then June really couldn't detect the green area or plant height that well. So when we're doing in the actual breeding, what we know is we want to know the trend. We don't really exactly know yield is 100 or 200. It's kind of maybe 95, it's good enough, almost like 110, right? So we want to know the trend. This really shows so the blue one is we measured by hand, the actual forage biomass of Sicer milk wedge. And then this predicted one is a red one based on drone data. And then you can see the trend is very similar. So that really means it's potentially a very good application to the breeding programs. Another thing is genomic resources. Well, a couple years back, or maybe 5-6 years ago, we have never thought forage we can develop this many reference genome. So reference genome is kind of a digital nucleic acid sequence database, or basically a database for the genes of a plant. And there's lots of human reference genome, right? So if something happens, we probably can, for example, myself, I sequence my gene and reference genome, and we can see, okay, I don't have this gene, or this gene is going wrong, and that's causing disease, right? So the reference genome is very useful in a plant breeding, and then now people are just crazy doing different things. Because of this new technology, it's much cheaper, and people can do it. So alfalfa, there are a couple of them underway, at least 4 as far as I know. Now people are doing meadow brome, which is almost done, Sicer milk wedge, hybrid wheatgrass, sand for intimacy, red clover, and European groups do red clover, perennial ryegrass. It's basically a kind of era of reference genome development. So it's going on. It's a new thing, a new trend, and going to be very useful for breeding. So one of the example of why reference genome is important is, I know it's a very technical thing, but I give you a very general idea. So now the new breeding tools is genomic selections. The genomic selection is not really using gene editing or genetically modified. It's basically based on all these genes. You can calculate out, for example, myself, if for example, a plant, let's assume based on my gene breeding values, this plant can grow 200 pounds and another plant 100 pounds. You potentially, based on your genomic information, go to the 200 one for high yield, right? So all this thing really helps reference genome and new breeding tools. So this is probably also important for forage varieties. This is some of work from my lab. When we have all these forages, people always said, you don't know whether that's original one or different one or 100 years ago. One of good example is Kentucky 31, TOFSQ. And I was in a conference 2 months ago, and then there was a person from Utah, the scientists from Utah actually demonstrated Kentucky 31 nowadays, they are not Kentucky 31. It's just the whole, on the map, it's just everywhere, right? So that's really happening on a number of different species, including probably red clover too. Some of red clover variety, you probably think that is the variety, but maybe it's already evolved to different genotypes or genetic. So another aspect for marker development is, molecular marker development is, you can actually identify for that kind of purpose. You can also protect the varieties. For example, if we really think more strategic way, you can probably genotype them, and this variety is this variety versus the other one. So that probably helps too, but it's maybe some consideration before we apply. Before I finish up, I talk about gene editing. Gene editing, I know some of you probably didn't like, or it's a more controversy. So I actually took most of information here is from Canadian Food Inspection Agency. It's the government regulations. Really, it said plant containing foreign DNA always require authorization. So you cannot release a variety if it's genetically modified. But gene editing technology, this is their word, gene editing technology can be used to make specific changes to the genome of a living organism, including plants, by adding, removing, or altering genetic sequence at precise locations. And gene editing tools can be produce similar or identical outcome to conventional plant breedings and doesn't require special authorization. So I know I'm not doing it, but this seems going to be quite a new directions. And I went in a plant and animal genome conference, and I was really surprised how much people had advanced in alfalfa. They actually use only 3 to 4 months to develop a gene-edited genotype. So it's true. So this really is because of CRISPR-Cas9 platform. It's a very precise editing, gene editing platform you potentially can use. So Dr. Stacy Singer provided me some slides. So this is one of, it was pretty detailed, but I won't go through all the details. So this is alfalfa again. All these gene editing forages are mainly on alfalfa right now. And this is one SPL8 genes, and then you use CRISPR-Cas9. I will only focus on the pictures here. The first one is the 2 weeks after they dried out all of them. It's basically trying to dry out or testing their extreme drought tolerance. The non-gene edited ones didn't recover when they providing adequate waters, but the other two edited at different levels, 50% versus 75%, they actually come back. So that really means gene editing, it's different, right? So this is one of examples. Also, Stacy provided me this slide. It's another genes. This was alfalfa tolerant to waterlogging conditions. So it's also, you can see well-watered 3 weeks after waterlogging. So it's kind of 3 weeks of standing waters and 6 weeks of standing waters. You can see gene-edited versus EV is wild type or original genotypes. You can see the difference, right? So there it's working a number of different ways. So Ali provided me another slide. This is another multiple genes editing, and then I don't go too much. And his really goal is flowering, delaying the flowering time and branching. So he's a kind of, you can see wild type and some sort of increased branching in a gene editing. So I know gene editing still requires lots of testings and all this thing, and I personally haven't done any, and then I don't have experience. I believe it's still, even it's not required authorization to register, still required to follow a number of protocols, and then all this is very important because if somebody wants to do more conventional thing, and when this comes in close by field, you potentially get contaminated by insect pollinations or some other ways. So as I mentioned, this is my last 2 slides. We really need to, the forage breeding program is shrinking. We probably need to work together. And this is one example. We are Beef Cattle Research Council funded project. what we are doing is, it's all conventional breeding programs. We all work together. Each program provides 1 or 2 best germplasm of alfalfa, and we are testing them in 6 different locations, and then we are resharing all the information again. So it's very easy conventional breeding, but when you multiply by 6, it's quite dramatic. So this is probably important to some of you or all of you. All this breeding program has to have a value towards the end, and then it's important to the industry provide certain specific guidance, what we really want next 10 or longer term, and then it's because it's a long term. Sometimes even we wants to do certain things, this funding model is also important. Annual crop have really good royalty models, but perennial crops, it's hard to rely on royalties. With that, I will thank all of you and then some funding agencies. Thank you.